evident from last 15 years that phase separation play a huge role in membraneless compartmentalization inside cells. But the interesting property is the macroscopic material property of the condensate can vary widely, and it based on functional demands, and sometimes if it is not controlled, it has a huge pathological implications. Now, while condensates can show extremely liquid like property like fusion, surface weighting and other things, they can also form gelation and amorphous solids, and on the other hand they can also form very structured and very ordered structures like amyloid. I have shown at the right side. So, on top of that, upon phase separation, liquid like condensate can also change its material property with time, and there are various proposed mechanism for that. There might be lot of polymer concentrated inside the condensate, so there can be a polymer entanglement, and because of that there might be amorphous solid formation. There can be physical cross-linking and network like structure formation. Because of that there can be percolation, gelation and lot of things. And sometimes, since a huge amount of protein is concentrated inside the condensates, the protein can now forced to go in the misfolded conformation. There might be a conformational change associated with the phase separation, and now certain interactions can drive for the fibrillation. If all these things are regulated by cells, then it is fine, and cell has actually utilized a lot of condensates which are not very liquid like, they are like solid like in nature, they have used for like storage purpose and other things. But if it is the process is not regulated, as I have mentioned, the fibrillation has lot of pathological connections, and it has already been seen several neurodegenerative associated proteins, they form fibrils. Now, I am interested about how hormones are sorted and stored inside secretory granules. So, hormones are a class of peptides, proteins. They are chemical messenger. They release into the blood with some chemical message when it is only required to the body. For example, now I am giving a talk, I may be very excited, so adrenaline and dopamine might be rich with those hormones, and when I will be sleeping, I will be rich with melatonin. So, when the hormone is required to secrete, then only it will be secreted, and other time it will be stored inside secretory granules in the cell, certain cell types called neuroendocrine cell, which has the machinery for the regulated secretory pathway. So, the cell has a basal like the default mechanism which is called constitutive secretion, where protein is synthesized and released. But certain cell types, for example neuroendocrine cell lines, they have the machinery which will package hormone from the transgolgi network inside a membranous granules, and which appears as a dense core when it is looked under microscope. So, these are prolactin granules, they form a dense core and they store for a long time. So, on the right hand side it is a readily releasable pool of the secretory granules, and when there is information from secretagogue, it will release that, and although they are in the aggregated state in the inside secretory granule, when it will be released, it will be released as functional monomer to serve the function. Now, the thing is how hormones are sorted from the transgolgi. Several previous reports have mentioned that they have aggregation properties. So, aggregation might be one of the mechanism for hormone sorting inside secretory granules, and my supervisor Professor Maji, he has done in one of his postdocs, he has seen that he chose like 42 different hormones, and he found like all of them has a property to undergo functional amyloid if a certain condition is given. Not only that, when immunohistochemistry was performed on pituitary tissue, it has been seen that hormones, they are actually co-localized with amyloid specific dye thioflavinase. So, which is kind of giving the information that okay, hormones might form like it is forming functional amyloid. So, now I ask the question like how aggregation and related to functional amyloid formation, how it is happening, what is the mechanism for concentrating hormone so that it can form aggregates. So, when we ask if phase separation can serve the purpose to concentrate it in a confined volume to promote the aggregation. So, I have chosen 10 different hormones and they are actually very different from their location. They are very different on the amino acid length, their molecular weight and their secondary structure, and I wanted to see what is happening. So, now, these are the 10 hormones: galanin, somatostatin, beta endorphin, calcitonin, neurotensin, glucagon like peptides, then leptin, growth hormone, prolactin. pH play a very important role in the regulated secretory pathway, because pH in the endoplasmic reticulum gradually goes down till trans-Golgi network, and inside the secretory granule pH even goes around 5.5 to 5.8. So, acidification is one of the major thing that happens in the secretory pathway. So, I wanted to check what is happening if I decrease the pH. So, I took all the hormones at a fixed concentration and then I employed to check what is happening in pH 6. So, out of 10 hormones, only 3 hormones I have seen undergoing phase separation. And then when I added glycosamine glycan, now glycosamine glycan is called like helper molecule. It promotes protein aggregation and it is also known to be present in the trans-Golgi network along with other metal ions. So, when I have checked the effect of glycosamine glycan heparin, I have seen all of them undergoing phase separation in pH 6. They have beautiful surface weighting property and fusion property. So, there is a video of fusion, you will see like neurotensin undergoing phase separation, and there is also surface weighting property which is showing very liquid like nature of the hormones upon phase separation. So, now I picked 2 hormones, growth hormone and prolactin. I recombinantly express and purify it for in large volume to further understand what is happening. So, these two, the advantage of these 2 hormones are there is no processing event throughout this hormone packaging. So, several hormones undergo processing like from the pre-pro hormone to pro hormone and other processing. These 2 small peptides, 191 amino acid and 199 amino acid residue, they do not have any processing event. They are very structured, there is hardly any intrinsically disordered region, as I have done in some in silico analysis to find if there is some intrinsic disordered region, there barely any intrinsic disordered region. They have some amyloid promoting sequence when I have analyzed with Tango. So, then I checked if they are undergoing phase separation, if I vary the concentration of both the protein. So, if I increase the concentration of growth hormone or prolactin with increasing GAG concentration, they undergo phase separation. But the nature of the phase regime of growth hormone prolactin is quite different. Prolactin mainly undergo phase separation when it is added in the 1:1 ratio, and with increasing the concentration of the heparin, it is undergoing some clumping of the condensates, while growth hormone actually separating very well with increasing GAG concentration. So, that means that although hormones are undergoing phase separation in presence of GAGs, does not mean that the interaction nature will be same for every hormones. Also, like we have checked like how heparin is distributed inside the condensate by labeled heparin, and we have seen like for prolactin it is mainly concentrated at the center of the condensate, while growth hormone it is distributed. Also, the size of the condensate is very different if we increase the GAG concentration. For growth hormone, increasing GAG concentration, size of the condensate increase, while prolactin it is mainly at the 1:1 ratio, they are more pronounced phase separation. So, then I have changed the pH gradually from 7.4 to 6. Now, in 7.4 without GAGs it does not phase separate, both of the hormones they do not phase separate much, but if I decrease the pH even without any external added things, both the hormones intrinsically undergo phase separation. And in pH 6 without anything in 200 micromolar around concentration, both of them undergo phase separation. And also we have seen like very sharp kinetics of the phase separation when we have performed light scattering as compared to its pH 7.4 phase separation kinetics. Also we have performed centrifugation assay to understand what is the dense phase and dilute phase concentration, and we found like in the dense phase the protein is concentrated in the millimolar range, and it is also known previously that inside the secretory granules also hormones are heavily concentrated in millimolar range. So, the phase separation might be one of the mechanism for concentrating a huge amount of hormone inside secretory granules. Then we further employed the effect of chondroitin sulfate, another GAGs, and effect of zinc metal ion to understand how the phase regime is changing with further addition of these two parameters in pH 6. And we have found like growth hormones specifically in presence of zinc, they have formed some clumping type of nature and clump condensation, while prolactin in presence of CSA they formed condensates, and the effect of clumping in presence of zinc is less in prolactin as compared to growth hormone. So, growth hormone has some kind of special behavior with zinc, we will see what is happening, and then it is so far so good that okay, they are undergoing phase separation. What is happening after phase separation? So, we have done FRAP experiment. So, what we have done, we bleach a certain region of interest, and we have monitored like how that region is recovering with time. So, it is one of the way to understand the translational dynamics of the molecule. So, if the molecular translational dynamics of the molecule is very high, the FRAP recovery will be very fast. If it is low compared to our detection rate, then the FRAP recovery will be low. And it is one of the way to understand okay, there is some kind of maybe structure formation or some network or some cross linking, something is happening, that is why the FRAP recovery is not like happening fast. So, we have seen like all the hormones at 0 hour compared to 0 hour, within 1 hour the recovery is very low. We have calculated the bromyl fraction at 0 hour and 1 hour, we have seen like all the hormones, their FRAP recovery is lower at within 1 hour. Then we further look in details what is happening for growth hormone and prolactin. This is pH 7.4 phase separation in presence of GAGs, and the initial recovery while around is 80%. In pH 6, the initial recovery falls around 40%. So, pH is promoting some kind of solidification inside the condensates, and it is true for both growth hormones and prolactin. So, we have checked the how this liquid to solid transition happening. Now, the next question is what is the solid? Is it amyloid? It is what is this? So, we have tracked with one of the amyloid specific dye thioflavinase of all the condensates, and we have imaged at 0 hour of phase separation and around the 12 hour. And we have found like all the hormones there is thioflavinase fluorescence enhancement, and which is very predominant for certain hormones. So, for example, galanin, somatostatin, beta-endorphin, their thioflavinase fluorescence enhancement is very sharp. And we have also seen like you will see that fibrillar structures are emerging from the condensates, and there are these 3 candidates which are showing very sharp thioflavinase fluorescence enhancement with time. And there are some other beautiful image of how fibrils are emerging from the condensates, and these are thioflavin TEM electron microscopy image of the condensates along with the aggregated structures. Now, this is thioflavin TEM images for 5 candidates, hormone candidates, and we have found that at 12 hours they are mainly the mostly aggregated structures. We have also performed FTIR to understand the secondary structures, and we have found like for example, for galanin that 1695 peak is associated with the cross beta structure, and you will see like with time it is increasing. Around 12 hours it is very much increasing, and all the like here the under study all the candidates they have that cross beta secondary structure which is shown by this characteristic peak of 1695. So, then we try to understand what is, because we have the control for growth hormone prolactin, we have huge amount of hormone, and we have found like it is phase separating in pH 7.4, in 6, also in presence of GAGs, metal ions. So, we employed all the individual conditions, and we have seen like the thioflavin T kinetics in presence of all the conditions. We have seen like although both the hormone undergo phase separation in all the conditions, certain specific condition is required for the thioflavin enhancement, and for growth hormone it is zinc which is showing the thioflavin enhancement, and for prolactin it is chondroitin sulfate which is showing the major thioflavin enhancement. Another study we have performed, we have done time dependent dot blot assay to understand like and we probe the dot blot with amyloid specific antibody OC and oligomer specific antibody A11, and we have found like from the very beginning growth hormone is showing OC positive, but it is negative for A11. So, they are actually associated with very fast aggregation, amyloid aggregation with no oligomerization, whereas alpha synuclein when we have studied, we have found it is alpha synuclein phase separated solution we have employed, and we have found like they are positive for A11, they have oligomeric population, but up to 12 hours there is no amyloid aggregation. So, it is showing like growth hormone is undergoing very fast amyloid aggregation in presence of zinc. We have also purified both aggregates from fibrils from both growth hormone prolactin in that specific conditions, and it is showing the XRD showing the characteristics peak for characteristics diffraction pattern which is equatorial and meridianal for like amyloid which is 4.4 angstrom and 10 angstrom, and also like we have seen under the electron microscope it has fibrillar nature. Here is also we have seen like 1695 peak in the FTR which is showing they have amyloid fibrillar nature. Now, after performing those in vitro experiments, we were interested to see what is happening in the neuroendocrine cell lines. So, I have mentioned that certain cell lines like has that special machinery to store secretory granules, and ATT20 is a mouse pituitary cell line which endogenously produce ACTH and beta endorphin hormone. So, I have done the immunostaining, and we have found like both the hormones are actually stored in the co-stored in the granules, and they are also showing co-localization with granule specific markers like chromogranin A. So, this cell is a good model to study for hormone aggregation. So, then when I have transfected transiently growth hormone with EGFP, so the plasmid construct has growth hormone, at the C terminus it has EGFP, so that we can monitor what is happening by EGFP fluorescence. So, when we have transfected it, we have found one population at the perinuclear location and one population at the cell tip. Now, perinuclear location, when we have stained with trans-Golgi specific antibody TGN38, it is co-localizing. That means that perinuclear location is actually located in the trans-Golgi region, and they are actually nascent granules, whereas the tip population, they are matured secretory granules and they are ready for the releasing purpose. And now, when we have only transfected with EGFP, there is no granule formation, there is no puncta formation, and whereas the growth hormone, when it is along the EGFP, there is the puncta formation, and that means that the puncta formation is very much protein specific phenomena. Other thing is when we transfected this in a constitutive pathway containing cell line, for example 3T3 fibroblast cell line, we have not seen any puncta formation. That means cell type is also very important to study the how this process is happening. So, these are the dynamic nature of the granules. You will see they are highly dynamic in the cell. So, this is the TGN population and the tip population, and also to understand the dynamics in the 3D volume, we are doing some lattice light sheet imaging, and it will be it will showing how dynamic they are inside the cells. And also we have found in the TGN fraction those condensates, those TGN fraction is highly packed with the condensates, and they are actually undergoing frequent fusions. Then we have checked that the granule formation at time dependent manner, like at early time point around 16 hours, then 24 hours and 48 hours. We have seen like with the time the granule size is increasing, that might be linked with like more accumulation of matured granules as compared to nascent granules. And also then we have isolated the aggregated fraction and the soluble fractions by treating the cell with lubrol. It is one of the well known method for secretory granule to isolate the aggregated fractions. We have seen like at early time point like 16 hours the aggregated fraction, which is the pellet fraction, and here it is noted as P, and the soluble fraction is noted as S. The early time point the soluble fraction is very high and pellet fraction is low, and with time that soluble fraction is low and that pellet fraction, that means the aggregated fraction, is very high. That is indicating that at later time point, in the since it is a transient transfection, so there is only one time it is treated with that plasmid, and then protein is not stably expressed, and with time there is more population of the mature secretory granules inside the cell. Another experiment we have performed, we wanted to see what is happening by changing the temperature. 5 minutes, yeah, I'll finish quick. So, previously it was shown, it was reported that at 20 degrees centigrade there is no mature granule formation. So, we thought that might be because of at low temperature the aggregation is now inhibited, and indeed we have seen like when the temperature is low, the lubrol soluble fraction is the pellet fraction is almost nil at low temperature. That means the aggregated fraction is there is not much aggregated fraction, and along with that the cell does not have the tip population. So, we have also seen that in 20 degree thioflavinase also binds less as compared to 37 degree. Another thing we have checked that what is the pH responsive behavior of the granules, because it has to be reversible to release that aggregated fraction in the cytoplasmic pH 7.4. So, when we treat the cell with chloroquine, which is known to increase the pH of the acidic organelles, we have seen like destabilization of the granules, and now there is more soluble protein fraction throughout the cell after chloroquine incubation. And also the in vitro study shows that when we increase the pH, the turbidity goes down, so phase separation is reversible with increasing the pH, and again it is when going back to 6 the turbidity is high. So, the phase separation behavior is also pH responsive. And now we try to actually differentiate the TGN fraction and TIF fraction by fluorescence lifetime, and we have found like that EGFP lifetime is less in TIF population, which is showing it might be in the aggregated state. Then we have actually isolated that EGFP fractions, and we have seen like it is positive for amyloid specific antibody OC, and also we have isolated the secretory granule fractions from 80-20 cell, and then we immunoprecipitated that EGFP granule fraction, and they are under the electron microscope, they are granules. We have performed cell cross section study, and in like when the cell is cross sectioned at 50 nanometer, and then we have found like they are forming that granules. And now this is one of the interesting study. So, now 80-20 or any neuroendocrine cell, they store that population for release, and that release happens when there is a signal from the secretagogue. So, we mimic that secretagogue mediated stimulation with BACL2. When we add BACL2, we have found that the tip population is vanished upon BACL2 addition. That means that portion is now released, and when we actually collected that media after the BACL2 treatment, and we have compared it with the basal treatment, we have found like upon the secretagogue mediated treatment, and that protein is actually releasing growth hormone, and we have actually quantified also that data. This is the last experiment. So, what we have done, we have now deleted that amyloid region from the protein. So, there are two amyloid region in growth hormone prolactin. One region is in the signal sequence, and signal sequence actually cleaved off when protein enters from enters into the ER lumen, and another protein amyloidogenic sequence is inside the protein throughout the process. We have systematically deleted that these two amyloid region from the plasmid, and we have checked like when that amyloid region from the signal sequence is deleted, that protein is pancellular, there is no granule formation. When that region is deleted from the peptide sequence, there is like here and there granule formation, but they are not like informed as TGN fraction and that cell tip fraction as we have seen before. So, that means that amyloid sequence has certain information with it which shows that it has to be formed granules and it has to be released in a systematic way. We have also done some in vitro study where we have seen that when we release that amyloid region from the protein is now in presence of PEG, it is actually in the surface, it is not a proper phase separated, like it is a hollow condensates. So which is showing like the amyloid sequence is very important. And now here I will stop, and I will thank my supervisor Professor Maji, and this is our beautiful group and we enjoy a lot. And this is there are other people who have actively worked, Punna, Prasad and Anindo and Sutapa. Thank you. I will happy to take questions here, please.
Sandeep. Okay.
I have a comment and a question.
Sure.
The comment is based on the slide in which you showed soluble and pellet and the population is inverting. If you could just go to that slide. I think about 15-20 slides back.
Yes.
Yeah. So look at those surface, the soluble and pellet bands.
Right.
Although the population inverts from 24 hours to 48 hours, but there's one thing that is seen uniformly, which is that the mobility is not the same. The soluble band is always a little above the pellet band.
Above, yes.
Right?
Yes.
Now, what that suggests is that there is some processing going on, and the polypeptide chain is not of the same size.
And we have actually observed it very consistently.
Yeah. So there must be, if you do some mass spectrometry,
right,
to see what's happening at the ends,
right,
there must be some N-terminal or C-terminal processing which is driving it towards the pellet.
That might happen.
That's the comment.
Yeah, thank you for that.
The question is, of what possible use is it for nature to cause the growth hormone to form amyloids in granules and then release those granules? What is the provision for the protein to then go back into solution and work from the amyloid aggregates?
So
how do they dissociate? Is it a pH dependent thing? I mean, is it being kept in an amyloid form at a particular pH, and when it's released it dissociates, or what is happening there?
So on contrary to the pathological amyloids, they are actually very responsive, like they are kind of reversible nature, and here also we have seen like with increasing the pH. Right now actually I am doing experiments where I have isolated that functional amyloid from growth hormone, and I am checking that with increasing the pH, how that functional change, like secondary structure change is happening. And yet we have observed that with increasing the pH, they are actually dissociated. Like they have that pH reversibility. And previously also in the 2009 paper, it has been shown that when it is like there might be two factors. One is pH, and another is dilution, because inside the secretory granule it is heavily crowded, heavily concentrated, and when it is released in the cytoplasm, there is a huge dilution. So dilution is factor number one, and another is pH, which actually makes it reversible.
So that's very interesting. So presumably from the liquid phase separated state also there is reversibility, and from the liquid solid phase separated state also there is.
Yeah. So
what is the kinetic partitioning like? I mean, why are there these two stages, and why do you observe both interconvertibly, and then there's a third interconvertible state, which is the soluble state?
Yeah, a very good question, but it is very hard to distinguish between the liquid and the solid, because this process is so fast and it's kind of a spectrum which is merged with each other, because within 1 hour we have seen like there are some hormones which have been showing that within 1 hour it has started to bind thioflavinase. So it is like phase separation very immediately, and we don't know how are they at all different process, or how quickly they are happening? What is the molecular mechanism for that? It is I can't comment on that. Yeah.
Okay, just on this video only. So you have these droplets. Now you saw that they were moving a lot.
Yeah.
Now, why are they moving? Like, is there some sort of a concentration gradient? Or, I mean, I'm just curious, is there some, and what kind of diffusion pattern do you see of the droplets? The second question is, do you see fission also, or you just see fusion?
Very good questions, and I think it is these are some aspects which we should look in, like in future. Right now I have not actually studied how the diffusion what is the diffusion property, but actually they are very much dynamic. So there might be some reason behind that, and this is an interesting aspect to look into the future. And fission, I have not actually noticed to look for the fission, and I'll keep that in mind.
So in the interest of time, let's thank the speaker, and let's welcome the next speaker, and Ayadurai from CSI CLRI. So I'll give you a head up at 25 minutes. Okay. Right?
அது ஆட்டோமேட்டிக்காக ஓப்பன் ஆயிரும், ஒன்றும் பிரச்சனை இல்லை. அதில் ஐ திங்க் ஸோ பார்க்கலாம். அப்படி தான் லோட் பண்ணியிருக்கேன். காப்பி வேண்டாம், ஏன்னா அந்த வீடியோ ஒர்க் ஆகாது இல்லைனா. Okay. Yeah, it's okay. So good afternoon to all. Thank you so much for the invitation. Probably my talk is really going to be entirely different with what so far you heard. Fundamentally, we are moving towards the basics to application science. So we specifically trying to produce or find the novel source for producing co-servates, that's a liquid separating proteins that could be used for basically for biometrical applications, for different something like tissue engineering, or basically which focus on wound healing development. So our topic is we basically working on biomaterials. Biomaterials, as you are aware, that those are biologically compatible, and it could be introduced into the body because it has a long biological compatible activity. And especially when you talk about the biomaterial, you immediately come to know that collagen, gelatin, or some elastin like proteins. Those are really good because they are biologically compatible, but still it has a lot of limitations. Especially we are much interested on developing some underwater wet resistant adhesive materials, because those kind of materials are frequently available or synthesized only by the chemist people. It has a limited level of biological compatibilities. So we are very much interested on finding and novel wet resistant biomaterials, because when we use those kind of materials, the water is a strong repellent, which never going to allow you to, it's not going to allow you to adhesive in any position. So we are really searching for some of the highly wet resistant material from marine and other sources. Some of the findings are it has been really reported that some of the marine organisms are generally produce some of the proteins. They are denatured, disordered proteins. They will undergo for a liquid separations that form a fluidic and protein rich condensed co-servates, and that hold the wet resistant mechanism, but that also preserve the dispersion of the proteins even in the aquatic environments. So we are very much interested on those kind of proteins, and we are looking on it we try to develop, we identify and we will recombinantly express, and we will try to understand the mechanism of how these co-surveys are really occur in the wet resistant conditions. One of the material what we are really working on is like barnacle inspired proteins and also the muscle inspired protein. Today I am just going to talk about some of the role and understanding the muscle food proteins on wet resistant proteins. So basically I think I'm not comfortable here, okay. So basically if you could see these muscles, probably everybody knows that when you walk on the seashore, the muscles are strictly adhered in the seashore, I mean the rocks. It produces some of the proteins called food proteins. Usually it produces the vessels, it produces the thread, and the thread containing n number of food proteins, basically from food proteins 1 and 2, 3 to it's go up to the variety of around 29 proteins. These proteins will go for a liquid-liquid separation among them, and it resulted in the condensed co-observations. Those co-observates usually help for the attachment, and it facilitate even they are very highly resistant even the high tidal. So when you look at that specifically, the muscle food protein 3 and 5 form very stable thermodynamically strong co-observates. That's look into the people are very much interested on it, and they still we exactly know the mechanism. Most of the time it has been proposed that these muscle food proteins are containing TOPA, that's one of the post-translationally modified amino acids from tyrosines play a very critical role on it. Because TOPA is a catapholic amino acids, it's a versatile amino acids. What's happening over there? When it get oxidized, what's happening? It's going to be a topoquinone. So it's highly reactive in nature. So it will always prone to go for a intra-cross linking. But in the case of reducing conditions, it will form a it's dehydrated. It will go for a very interesting hydrogen bonding interactions as well as metal coordination, metal oxide coordination. But as I mentioned, the TOPA oxidation is play a very critical role on wet desiccation. When it's going to become oxidized and it form a topoquinone, it prone to just release from the attached surfaces. So the questions here is how the seashore is, as we are aware, that sea is thermodynamically oxidative conditions. So at these conditions, how these muscle food proteins are really maintaining the strong adhesive nature is still unknown. One of the recent study, one of our collaborators reported that when they introduced this TOPA molecule on it, and they observed that it go for the intramolecular interaction between the TOPA that specifically hold the adhesive nature in the water. But still exact mechanism is underexplored. And most of the time when we try to study the muscle food proteins and related to the adhesiveness, all the studies are mainly focused on TOPA or TOPA based chemistry. We call that TOPA paradigm. Because based on the theory, lot of TOPA based material has been proposed and a lot of interesting adhesive material has been developed. That's very good. But what here is not unexplored is these proteins also have several other amino acids like phosphoserine and hydroxyproline and hydroxyarginine. These proteins also present almost like 15 to 20%, but still it is not explored. Even recently one of the studies very clearly informed that some of the hydroxyproline that present in the similar kind of co-surveyed forming protein, slime protein, and they play a very important role in the adhesive interactions. And we hypothesize that because as you also aware that the hydroxyproline is play a very critical role in the collagen. It maintains the triple helical structure, stability, and also play a very critical role on the intracellular assembly, the fibril formations. So we hypothesize that whether the hydroxyproline may play any role on in the case of co-observation formation by helping hydrogen bond formation or in the case of dehydrations. But the question here is why so far people have not tried to explore that one? Because one of the reason could be we don't have a specific methodology to understand or introduce such a kind of post-translational modified amino acids into the protein itself. Even we could introduce site specifically, or even we could introduce only one amino acid, post-translational modified amino acid itself become very challenging, because we have to go for enzymatic reactions to study all the modification, which is very difficult to control. And moreover, this case we have to introduce minimum two post-translational modified amino acids to the protein, then only we can suggest something. So here that's a major challenging and the biggest challenge for us when we started studying such an experiment. So because why even synthesizing through different approaches may not work here. We need bulk amount of samples to mimic similar kind of study the bulk adhesiveness to propose or understand more about the conservation mechanisms. So we have in our lab, we developed a new approaches. We are working on this for several years. What we are trying to see here is we introduce unnatural amino acids, that's a post-translational modified amino acids in any amino acids into the protein through either two ways. One is we use genetic code engineering approach, where we try to misaminoisolate the amino acylation there instead of our target amino acid. For example, if I want to introduce proline, we completely deplete the proline in the medium, and we will apply and we will introduce a hydroxyproline in the system. That what will happen, our proline tRNA and tRNA synthetase will recognize, and it will go and introduce through misaminylation. Then second one is, we are also working on that one, but the first one is the major challenge would be, it's going to be random. I mean, it's going to be residue specific. Wherever the proline will be there, it will be replaced, and we cannot control site specifically. But we also have another method. We develop usually orthogonal tRNA and tRNA synthetase system, which can specifically recognize our unnatural amino acid, and it will be going and introduce site specifically at particular position. For example, if you want to introduce at some of the active sites, we could introduce site specifically by developing orthogonal translation machinery. So in our lab, the first one, what we developed, the first important thing is we overcome the challenge. We develop single and double unnatural amino acid incorporation. What we did is, like, we selected the one of the genetically modified E. coli, that's the oxotroph system. We have optimized the system to introduce proline, hydroxyproline instead of proline. And we also developed a multi-oxotrophic system where we have introduced topo and hydroxyproline simultaneously in the systems. And we observed that after that whether the system is really going to form a co-observation or not. We induce a co-observation conditions. And where you could see here, very interestingly, when proline alone, we could observe only the aggregation. It's not going to form any kind of co-observate micro droplets. But in the case of hydroxyproline induced or the TOPA, or even the multi-unnatural amino acid incorporated co-observates, we could clearly see the micro droplet formation, which is one of the best indicator of the real co-observation, the liquid separation occurs. So we studied them further to understand. We collect the samples and we centrifuge and we collect them co-observates, and it's subject to the fluorescent analysis. We treated with thiafluorine, and you could see here when we introduce two unnatural amino acids, 5 times increase in the fluorescence. It's almost like compared to the other the fluorescent intensity was very high. So it's clearly indicate that when we introduce two unnatural amino acids, it go for a crosstalk and increase the co-observation efficiency. And also when we analyzed with different pH the role, because we just mimic the C conditions, we treated with the different pH, and also we treated with the different kind of salts, and we could observe that at the level of 6 to 8, the double unnatural amide, I mean when the protein having TOPA and also hydroxyproline, it goes a very good separation over there. When we study the phase diagram, and we could notice that when we have two unnatural amino acids, I mean TOPA and hydroxyproline, the amount of protein required for co-observation formation is very less. So compared to the TOPA, I mean earlier everybody believed that the TOPA is really play a very important role. But here we could see that the two unnatural amino, I mean hydroxyproline containing co-observates also facilitate in the co-observation formation. But one thing, as I mentioned, TOPA is very good in the case of the, it's really help and facilitating the wet adhesive nature. But the question comes here is, what will happen when the oxidation happen? Whether it's going to completely disperse the co-servate mechanism at which all the hydrogen bonding will be go off, and it will disperse the co-servation formation. So what we did is, we have treated, we treated the protein with in the oxidation condition, that's sodium perrhyate, and we observed that here you could see very clearly when the muscle food protein, where we do not have any post-trans modified amino acids, it's still very stable because it's not going to form any co-servates, still aggregates or the aggregations are maintained. But in the case of proline loaded co-servate, still after oxidizing it retains the micro droplets. But in the case of TOPA containing co-servates, very interestingly you could see that after once it get oxidized, it's completely lost the co-servation pattern. You could see the micro droplet has disappeared. But in the case of two unnatural amino acids, I mean where the protein encoded with hydroxyproline and TOPA, still it maintains the co-servation efficiency. And in order to make sure that whether it is really to support further, the proteins were we study the electropotential of the proteins by cyclovoltammetry analysis. You could see there before oxidation, the TOPA containing protein containing a good level of electropotential. But after oxidation, you could see here the TOPA containing proteins and the hydroxyproline almost lost its electropotential. But very important things what we have to observe here is the two unnatural amino acid. I mean when the protein having hydroxyproline as well as the TOPA, it retains almost 50% of the potential capacity. So it clearly says that the formation of the hydroxyproline, I mean introduce the hydroxyproline may form the transition peptide bonds, which may facilitate or it may really help to maintain the tautomeric equilibration of TOPA during the oxidation conditions. And we also studied the oxidation potential and also the co-servation formation in the case of bulk adhesive nature, because really it maybe works in a very least amount, but we have to make sure that that kind of bulk adhesiveness is really happening over in the system also. Unfortunately, the video is not running here. So we have observed very clear pattern that when it get oxidized, the TOPA, I mean the hydroxyproline containing co-servates are converted to topoquinone. When it become converted as a quinone, it lost its co-servation ability. However, when in the case of proline containing hydroxyproline containing amino acids are loaded with the system, so somewhere it supports it, and especially it facilitates to maintain the original characteristic feature even in the tidal oxidative environment. But the question again comes here that everything is okay, but the problem is really the hydroxyproline is play a very important role, and can you confirm it again by any other approach? So what we did is we have instead of hydroxyproline, we have introduced the butylproline. That's one of the hydrogen breaker, the hydrogen bond breaker. When we introduce those unnatural amino acids instead of hydroxyproline, and we could clearly see that all the proteins are lost that co-servation formation. We couldn't observe any kind of micro droplets over there. So it's clearly indicate that our hydroxyproline is really facilitating the formation of co-servation at the oxidative conditions. And we also studied the adhesive pattern using AFM, and we could observe a very clear-cut pattern that when we have two unnatural amino acids only, we could maintain the similar kind of bulk adhesive nature. And our bulk adhesive natures also confirm that when the shear strength of two unnatural amino acid containing proteins only have the ability to significantly show the higher level of adhesive nature compared to the single one either with the hydroxyproline or TOPA. So we hypothesize that. Finally, the studies, what we conclude is, when the TOPA alone, it could be fine. It could efficiently go on bind with the different surfaces. But when during the oxidative conditions, still it exactly, we almost exactly know that what exact mechanism going on in the surfaces when the plaques are producing different kind of proteins. But we could at the moment conclude that, or some kind of suggestion we could say that the hydroxyproline or other hydroxylated amino acids also could play very important role similar to the TOPA. It could help to maintain the oxidation potential, although it has been reported that when muscle food proteins also produce or co-segregated with a lot of antioxidants to maintain the oxidative potential in that particular environment. And also similar kind of amino acids or the hydroxyproline, hydroxyarginine to the hydroxylated amino acids also have play a very important role. So we could give a equal priority that such a kind of modification also should be explored. So the ultimate idea is to create what is the use of creating or understanding the co-surveys in our cases. Like we try to use those kind of co-surveys to mimic like a extracellular matrix to prepare your organ-like structures like organoids. So those kind of material could be useful to study, say, like an animal model. Maybe we try to replace them with like a kind of animal model. So we use these kind of co-surveys as a ECM matrix compared to collagen and other cases. So we explore the possibility of our co-surveys to prepare different kind of spheroids in our lab. So compared to other naturally using collagen, our co-survey also really supports our hypothesis that it wonderfully form a very good spheroids. So these are some of the co-culture pattern, and just show the applications, real applications of our co-surveys in the formation or different tissue engineering applications. These are some of our publications, and this mainly the work has been done by one of my graduate students, Cecilia, and also now partially doing with Sonam. So especially these, all the work has been completely done by our lab group member. Thank you so much.
Yeah.
At the end of a talk that I'll give tomorrow afternoon, I'm going to propose that the nature of the hydrogen bonding that exists inside an LLPS condensate is fundamentally different from what exists outside. And the reason that proteins by themselves under conditions of macromolecular crowding or DNA, or proteins with DNA, proteins with nucleic acids, the reason that they form LLPS spheres is basically correlated with their ability to hydrogen bond with water. So I find it deeply satisfying that you not only showed that hydroxyproline and dihydroxyphenylalanine, when they are introduced, the ability to form coacervates is increased, but you also said that this might happen with other hydroxyl group containing amino acids also.
Yeah.
This might very well be the case. In fact, I'm also going to plug the same hypothesis tomorrow afternoon.
Okay.
In fact, serine containing, threonine containing proteins with a very high content of these on the surface might actually show a greater tendency to coacervate with lower CSAT values and so on.
Yeah.
So I believe what you're saying, and I think you demonstrated it very nicely by showing that when you form the quinone, you lose the ability. So basically what you're losing is hydrogen bonding with water.
Yes.
You're losing the ability to reorganize the water in the droplet.
Yes.
Yeah.
So that's a major challenging. Whenever we people are trying to use TOPA-based chemistry, the question arises here is what will happen when TOPA get oxidized? Because it has a strong tendency to go for intra-cross linking. And for example, during the reducing condition, it will very well coordinate with metal oxides and even transition metal ions. But when it become converted as a quinone, so it lost all the ability. And we could observe here also that when it converted as a topoquinone, the co-rate also disposed. So the question again is, what's really happening over there? Okay.
Loss of the ability to have
Formation, yes.
Yeah, a fantastic talk. So I was wondering that, do you know anything about this? Because there are also a lot of people working with this calcium or metal and coordinated dopamine polymers, like Frank Caruso in Australia. And so I've never seen, they are actually talking about liquid-liquid phase separation. And I don't know whether they really didn't care about the fact.
No, the main thing is the interesting fact is this chemistry, what you are talking about is.
Yeah,
Actually, the TOPA is really fantastic material. Actually, it will go for a variety of chemical reactions. So people, like as you said, they will use it for calcium, maybe it go for a metal coordination. Yes, TOPA.
Or iron through too, I guess.
Yes,
with
yes.
Fe2, yeah.
Even frequently people use the topoquinone instead of TOPA also.
Yeah.
Because everybody use, if you look at very carefully, they will control the pH.
Yeah.
But most of the time, any TOPA based material, when it become reach around pH around 7.8, the question arises here, how is it really going to be stable? But somewhere it's good, somewhere it is problematic. Like we also did some work that we conjugated with some of the drugs, like boronated drugs. It will very well coordinate with it form a boron ester formation, but when it reaches the pH of 7.8, it will dissociate, the drug will be released. So you can also use it as a carrier, and you can use it for a controlled, a pH dependent drug release also.
So with that, let's thank the speaker once again. We break for coffee now, and we'll reassemble at 3:30.
A small announcement. For those of you
Yeah,
presenting
thank you.
posters, you can set it up right now. So the volunteers will be able